Now let us talk about the transformation. As I have already said that the transformation is the transfer of the genetic information through the agency of the free DNA. Here you can see in the picture, in the bacterial environment there is a naked DNA present. So these are the naked DNA. So these naked DNA are taken upon by the recipient bacteria. So this is the recipient bacteria that take up the naked DNA that is present in the environment. So the naked in the, DNA is now present inside the bacterial cell. So this after the bacteria take up this naked DNA, this um, naked DNA, we also call it is the donor DNA. This donor DNA gets incorporated into the recipient DNA. So here you can see the new combined DNA, that is the both there is presence of the recipient DNA and donor DNA, and the remaining there is degraded uncombined DNA is present inside the bacterial cell. And now I'm again going to tell you that the transformation is the transfer of genetic information through the agency of the free DNA. That means you can say that the competent bacteria can bind and import short pieces of environmental naked bacterial chromosomal DNA, usually from the bacterial cell lysis. And the transfer and the expression of the newly transferred gene is known as the transformation. So after this donor DNA, or we also call it the naked DNA that is present in the environment, gets incorporated into the recipient DNA, and that genes are expressed in the bacteria. This is known as the transformation. So these features, the feature of transformation is usually shown by the various bacteria like Streptococcus pneumoniae, Haemophilus influenzae, which is type B Haemophilus influenzae, and Neisseria group of the bacteria. This shows the transformation. And what we can do is that we can add the deoxyribonuclease, which degrades the naked DNA, and it prevents the process of transformation. So basically the deoxyribonuclease prevents the transformation in the bacterial genetics. So again I'm going to tell a brief about the transformation. So the naked DNA that is present in the environment of the bacteria is present from the bacterial chromosomal lysis, or also you can say that the competent bacteria can bind and import short pieces of environmental naked bacterial chromosomal DNA. This naked DNA comes from the bacterial cell lysis, and the transfer and the expression of the newly transferred gene is known as the transformation. So now let us discuss next about the conjugation.